Candida albicans is a normal component of the human microbiome, but overgrowth can lead to a fungal infection called candidiasis, which is more commonly known as a yeast infection. Candidiasis can be treated by a class of antifungal drugs called azoles. However, over time, some Candida strains may develop altered drug responses, such as drug resistance or drug tolerance. Drug resistance, which you may have heard of, refers to an increase in the concentration of drug required to inhibit fungal growth. Drug tolerance, on the other hand, is defined as the proportion of a population that can grow slowly, even at drug concentrations that would inhibit the rest of the population. For this project, I aimed to evolve an altered drug response in C. albicans by growing 8 different strains in media containing posaconazole, which is a relatively new member of the azole family. Every 72 hours, biomass was measured and a fraction of cells were transferred to fresh media. This was repeated 5 times. There were 2 general responses to the drug, which are shown in Figure 2. For the strains in the top panels, the majority of replicates just went extinct. The strains in the bottom panels, however, showed an initial decrease in biomass production, followed by some recovery by the time of the final transfer. The next phase of the project involved measuring resistance and tolerance in the evolved strains. Both traits can be measured using a disc diffusion assay, shown at the bottom left of my poster. If colonies can grow closer to the drug-infused disc, then they exhibit increased resistance. And if a larger proportion of colonies grow inside of that zone of inhibition, then they exhibit increased tolerance. The results of the disc diffusion assay are shown in Figure 3. An interesting trend was observed where strains with the lowest ancestral tolerance levels displayed the greatest improvements. Replicates from strain SC5314, which showed the greatest overall increase in tolerance, were sent for whole genome sequencing. After processing the sequencing results through our lab's bioinformatics pipeline, I manually searched each genome to find candidate mutations that were present in a large proportion of the evolved strains, but not the ancestral strains. 5 of these mutations were in genes that have a known association with azole drugs. This concluded my project. I was able to show that tolerance and resistance are separate traits that can evolve independently of each other, and I identified mutations that may be related to posaconazole tolerance. While these are exciting results, they also open new questions. For example, can high tolerance end up leading to high resistance? Additionally, does tolerance to posaconazole result in cross-tolerance to other azole drugs? I'd like to end this video by thanking you for taking the time to hear about my research and wishing best of luck to all the other students entering the poster competition.